biology, and I teach every once in a while intro bio. I teach the first semester of a 2-semester biology. By the way, if you are thinking of going into a biological science, you should most definitely take 2 semesters of biology and 2 semesters of chemistry, because anywhere you go, that's what they're going to want. And we have it here, and it's cheaper here. And it's good, too. So, just throwing that out there. All right, so, and if I had my way, you would have taken that before you took this. But nobody listens to me around here. Biology 1 and 2. Chemistry 1 and 2. Yep. Before you took this class. You would have been much happier campers. But anyways. So, peripheral, that's another good one. Peripheral nervous system, what is it? Peripheral, wake up! Hello? I had to use the microphone at least once today. Don't read the PowerPoint. What is it? Everything from the outside central nervous system that we just talked about in Chapter 12. This is the outside stuff, peripheral nervous system that feeds information into the central nervous system. I think I have to start drinking coffee again. So, sensory receptors are what we're going to talk about. Peripheral nerves and things that connect sensory to motor association ganglia are what we're going to find here, as well as remember that information coming out to cause an effect in motor. So we look at that same little crazy diagram we looked at earlier, and now we're looking at this portion of the diagram in this chapter. So the peripheral nervous system has both sensory and motor divisions. When we talk about motor or efferent, that also has 2 divisions. Things that happen automatically and things that I'm kind of aware of and have control of. The autonomic division of the peripheral nervous system also has 2 divisions: slow things down, speed things up. Which one's which?
Speed things up.
Sympathetic, speed things up. Parasympathetic, slow things down. So basically that's what this chapter is going to talk about. Some of the different receptors associated with the peripheral nervous system. So on page 484 in your textbook, they start talking about sensory receptors. Now, stimuli comes in many different forms. And we've kind of talked about them throughout the class, but now we're going to define them. So in order to classify these neurons, they're classified by what they're stimulated by. When I say stimulus, what do you think of? No, not what the stimulus is. When I say stimulus, what should pop into your mind immediately, because you've been studying Chapter 11 and you haven't forgotten about it? When a neuron is stimulated, what happens? What's that fancy term we use to say a neuron has been stimulated? Action potential, nerve impulse. We're going to start a track of message. Yes? So different neurons are stimulated by different stimuli. If I took a flashlight right now and shined it in your ear, would you hear better? Say no. No. Why? Because those receptors in the inner ear are not stimulated by light. Yes? If I screamed in your eyeballs, would you see better?
Maybe.
You might open your eyes a little bit, because I'm screaming in your eyeballs, but you won't see any better. You getting the point? Good. So, classification of stimuli is what they talk about. We have mechanoreceptors. What are they stimulated by? Some sort of pressure. Yeah. Mechanical stimulus, mechanoreceptors. Touch, pressure, vibrations, stretching, mechanical functions. Fire these guys off. Thermoreceptors. Temperature changes. Now remember, you don't have all of these guys everywhere. I'll give you a hint. If you want to pretend that you're very, very tough to a little child, tell them to come over and take this portion of your body and pinch it as hard as they possibly can. Go ahead, you do it right now. Pinch it real hard. Pinch it really hard. Doesn't hurt. If you do that here, you're like, ow. But if you do that here, nothing. How come? There's no receptors there's no mechanoreceptors and no pain receptors there. You can pinch the living daylights out of that thing and it won't even hurt. Will you get a bruise there? Yes, you will. Will you damage tissue? Yes, you will. So you might get a little bruise on your elbow if you pinched it really, really hard. But if there are no receptors there, I don't know that's going on. Photoreceptors. Light. So light waves, and when we talk about special senses next semester, at the beginning of next semester, we'll talk about these guys specifically. Chemoreceptors are going to respond to some sort of chemical change in the liquid that surrounds the dendrites of these receptors. Remember, the dendrite is where I'm going to start getting all these inflow of sodium. Remember, we can't forget chapter 11. That'll cause the nerve impulse to happen. So chemical changes, and we'll talk about your sense of smell. Yes, your sense of smell is a chemical sense. When you sniff the air, you bring in smell molecules, I call them, and they dissolve in a liquid. Mucus. I know it's gross, but it's important. And the amount of molecules that dissolve are going to stimulate the dendrites of the receptors in your nasal passages. We call them olfactory receptors. And if I cause enough of a stimulus, what's going to happen? Action potential, nerve impulse, send messages to the brain. Your sense of taste, also your gustatory receptors on the surface of your tongue in taste buds are also a chemical sense. So chemoreceptors. And then those pain guys that are going to pick up on damaged tissues. When I damage cells, think of me taking the cell, banging it on the side of the table, and opening it up like an egg. All of the guts spill out. And all of the chemicals inside cells are what's going to set off nociceptors. So they're going to pick up pain. So extreme heat, extreme cold, excessive pressure. Do I have any nociceptors in this portion of my? Nope. Also, inflammatory chemicals will set these guys off. So when you cut yourself and inflammation takes place, that's why sometimes pain is associated with them. Then we classify by location. Outside what? Exterior. So exteroceptors are going to respond to stimulus outside you. Yes? So the light, the cold, the pressure. Interoceptors or visceroceptors are going to respond to what? Things that are going on where? And give me a scientific name for all those organs inside your body. The viscera, visceroceptors. Whoa, almost fell. I'm having problems with my proprioceptors. I know, and there was a decaf in there. Wasn't even anything good. So, again, we're going to respond to what's going on in the viscera, visceral organs. Proprioceptors are going to help me with what? What I just almost had a problem with?
Balance.
Balance. Where I am in space. That's important, so I don't fall on my face. So it informs the brain about movements, going downstairs, going upstairs, and helps to coordinate joints and ligaments and all of those guys to help that movement take place properly. Classification by receptor structure. So this is another category in which we classify some of the different sensory and receptors that we're talking about. General senses: tactile sensation, touch, pressure, stretch, vibration. And then we have special senses, and again, we devote a whole chapter to those guys next semester. Your sense of everything that kind of takes place here in the head region: vision, hearing, equilibrium, smell, and taste. So we can have, if we look at table 486, we can see these guys. And again, these tables, take advantage of them, because what are they going to help you do? Make big information small. We want to do that, right? So we can have either non-encapsulated or free endings or encapsulated. So when we talk about epithelial cells and connective tissue, we see a lot of those non-encapsulated free nerve endings. When we talk about the integumentary system, we're going to meet a bunch of those that hang out in the dermis of the integumentary system. Some of them are non-myelinated, small diameter. We call them group C fibers. Things like temperature and pain and pressure, like touch, is what's going to get those guys rolling. Thermoreceptors, temperature receptors, can respond to different temperature ranges. And again, we have separate receptors. Some will be fired off by cold temperatures between 10 and 40 degrees Celsius. Celsius, not Fahrenheit. What's freezing in Celsius? 0. What's boiling in Celsius? 100. Heat receptors, we start firing those guys off at 32 to 48 degrees. They tend to be a little bit more deep in the dermis. Again, when we talk about the integumentary system, we're going to see it has two portions. The outer portion is called the epidermis, and the deeper portion, where all the blood vessels and nerves and all of these guys are, is called the dermis. So, nociceptors, also unencapsulated dendritic endings. They're going to respond to changes in surrounding areas caused by cell damage. So ion channels are going to open by heat. We also have pH detection, chemical changes. Capsaicins can also fire off nociceptors. Where do you find capsaicins? Yeah, and if you look at, if you're into hot foods, there's a rating on capsaicin level. What's it called? The Scoville units, right? And that's a measurement of capsaicin presence and how hot, how much pain you're going to feel, basically, when you eat. Exactly. And it has to do with these receptors and how they're fired off.
Is there a reason why, like when I was pregnant, I couldn't get it hot enough? Like the food?
Yeah. No, your taste changed quite a bit when your hormone levels are all over the place.
So do your receptors perceive differently, or is there other things?
You just have a. Why do some people like hot foods and other people don't? Yeah. Yeah. So it has to do with your chemical balance, your likes and dislikes. So Light touch receptors, we'll talk about, again, when we talk about the integumentary system, we'll talk about things like Merkel discs and hair follicle receptors. If I pull my hair, it hurts. How come? I must have some receptor there for either touch or pain, right? And it's wrapped around my hair follicles. So we see some of these guys outlined for us in that table 13.1. Then we have something called encapsulated. So we have unencapsulated, encapsulated. Tactile receptors for more deep touch, not that surface pressure touch we just talked about, are called Meissner's corpuscles. Pacinian corpuscles, more deep pressure. Ruffini's endings or bulbous corpuscles, again, deep pressure. Muscle spindles, they're going to help with movements. So when I pull on them, it's going to help contract certain muscles to help stabilize a movement. Tendon organs, where am I going to find them? In tendons, exactly. And what's a tendon? Connects muscle to bone. So when I contract a muscle, it's going to pull on those. They're going to send information to the brain so the movement can take place. And joint kinesthetic receptors in what? Joints are going to help position and movement. Again, we see these guys. Oh, wait a minute. I told you the wrong thing, didn't I? The other ones, the unencapsulated ones, are on page 485 and 486 we find these guys, the encapsulated ones. So these receptors are all going to help us react to our environment appropriately so that we can survive. So survival depends on our sensation, our perception. Sensation is defined as the awareness of changes in internal and external environment. Are you aware of every single stimulus? You better hope not. Because if you were, you would go crazy. So perception is the conscious interpretation of those stimuli. So you aren't aware of all the stimuli you are getting all the time. Remember, we have areas of the brain that are going to help filter out some of that stuff for us. So, sensory integration, getting everything together, is important, and all of these receptors, exteroceptors, proprioceptors, and interoceptors are going to help play a role in relaying information to be processed for reaction. So, receptor level, circuit level, and perception level are the different levels of information that are being sent to the central nervous system. The receptor level is where? Yeah, so it would be on the exterior portion of that central nervous system, right? But then we have to have some sort of information that's being sent where? Yeah, we have to go process that information in the central nervous system. So the circuit level is where we're going to now hand that information off and send it ascending for processing at the perceptual level, which is where? The brain. This is where perception then takes place. So if I have no way of getting that message to the perceptual level, I'm going to have a hard time being aware of any sensation that I'm receiving. Does that make sense? Kinda? So this is sort of the level in which information is sent from the peripheral nervous system to the central nervous system. So receptors have very specific or specificity to stimuli. One receptor, one stimuli. Maybe two. But again, if I shine a light in your ear, you're not going to see better. The pathway that the receptors are going to take, in general, follow with stimulus, then some sort of afferent neuron, which will then cause an action potential. When we talk about processing at the receptor level, we have some sort of stimulus and some sort of sense organ. Then we have potentials, graded potentials. Some will fire off, some won't, unless the stimulus is what? Very, very intense. So if I go over and I pick this up, I'm going to need a certain amount of stimulus to do that, yes? But if I go over and pick this up, same movement, I'm going to need a lot more what? A lot more message, a lot more muscles to contract, a lot more stimulus has to take place. So that's going to affect the amount of neurotransmitter that is released depending on the amount of stimulus you're receiving. And neurotransmitters generate what we call graded potentials, which we talked about in Chapter 11 in sensory neurons. After a while, when I'm firing a neuron off constantly, if I kept doing that for long periods of time, it might actually cause damage to the neuron. So it's nice if the neuron could sort of take a break. Neurons have the ability to adapt their sensitivity to stimulus. So remember way back in Chapter 11 when we talked about the receptors on the surface of the dendrites that cause this whole thing to start the ball rolling? They will actually become less responsive to stimulus if they're stimulated consistently over time. In order for me to stimulate that receptor again, I'm going to have to up the stimulus. I'll give you an example. This morning, anybody put on perfume? Little spritz, kind of smelt it. Ooh, smells good. And like 2 minutes later, if you're anything like me, you can't even remember if you put it on or not. No, because you really don't smell it anymore because of the phenomenon of adaptation. In order for me to smell it again, what would I have to do? Spritz. Don't spritz again. You're good. For those of you who tend to spritz a little too much, you're good. Even though you can't smell it anymore, everybody else can. Okay? So that's what adaptation is all about. That happens to pain receptors sometimes too. When you've experiencing pain for a long period of time, sometimes you become numb to that pain because of the adaptation. In order for me to get it fired up again, I go jab you again. Say you get a bruise or something. If I touch you again and make it more painful, it'll stimulate those receptors again. So adaptation is a very important protection mechanism for our neurons. Phasic or fast adaptation receptors are going to signal beginning and end of stimulus, so touch, pressure, smell. And then we have tonic receptors that adapt very quickly. Nociceptors, pain receptors, and proprioceptors go through adaptation very quickly. Processing at the circuit level. We have several different orders of neurons. We have something called the first order neuron. That's like the first group of neurons that are firing off, conduct impulses from receptors like your touch receptors and your photoreceptors, and then send that information where? Remember, we're talking peripheral nervous system now. It's going to start sending to spinal cord, that first first group of central nervous system neurons. Second order neurons are then going to take that message and do what? Hand it off. Remember, now we're in the spinal cord. So where do I have to go now with my message? I have to go up the spinal cord. I have to get relayed someplace in those midbrain regions, and I have to hit the file cabinets, right? So that's my second order neurons. The guys that actually process or pass that information to higher cortical brain regions are what we call our third order neurons. So they're then going to relay that message and in and around those central brain regions, the thalamus, to what we call the somatosensory cortex, so we can get processing that sensory information. Then, once we get up there, we're at the what level? Now we're at the perceptual level. We've opened the file drawer. We're getting the files out. So now interpretation of sensory input depends on that specific local target. Where did that information come from, and which file cabinet are we opening up? So we have perceptual detection. The ability to detect the stimuli requires information gathered from several different groups of nuclei or summation of impulses. Magnitude estimation, how intense the stimuli is. That has to be processed as well. So me picking up the paper versus me picking up this really heavy sack is going to be falling into this magnitude estimation category. And then spatial discrimination is also at the perceptual level too. So patterns of stimuli can be identified when we do something called the 2-point discrimination test. And we're going to basically torture you in lab after the week you dissect your brain. We're going to do some little torture things in lab, which are oh so much fun, with temperature and pressure and poking and stuff. But if I take something that has 2 points and I start them very close together and touch you, it feels like one thing is touching you because you can't sort out that information. But if I start separating at some point, you're going to start feeling 2 separate things. So spatial discrimination is also very important at the perceptual level. Some of the main aspects of sensory perception, abstract properties, quality discrimination, and pattern recognition. We use all of these different things to just discriminate things in our everyday environment. You know, when I look at a picture, when I look at a face, when I look at a group of things, there are many different things in what I'm looking at. And all of these work together to help me work out the stimulus that's coming in. When I look at a chair, or a group of chairs, or a group of people, or different features on someone's face, all of these guys have to work together in order for me to perceive the stimulus. So again, we see this diagram of the different levels of message and where they have to go for us to actually perceive what's going on. Perception of pain is talked about a little bit in the book as well. And people have different levels of pain perception. Again, that's because we're all wired different. If I walk up to somebody and give them a little tap, they might feel lots of pain, whereas somebody else is like, didn't even feel that. That has to do with the way you're wired and your perception of pain. So, warns of actual impulses that are damaging tissue is what pain is for. It's a protection mechanism for you. If I can't feel pain, that can be very dangerous, can it? Because I could be causing, from external environment, some serious harm to my body and not even know it. So people that can't do this it can be very dangerous. And are there people that can't do this? Yes. Yeah, and there's been different shows and
Guys getting shot, cops wondering why.
yeah, that it's not affecting him at all because he can't feel the pain associated with it. So these are some of the impulses that travel and the pathways associated with perception of pain. So if I'm off, chemical imbalances can cause different types of pain perception as well. Sometimes if your body chemistry is imbalanced, you feel pain everywhere versus when it's in normal range. So all perceived pain at the same stimulus intensity. Pain tolerance, though, is going to vary. Some people are more sensitive to pain. They're said to have a low threshold of pain tolerance. Genetics might play a role in that. Pain medications, if you can stay away from those, stay away from those, because so many of them cause dependency issues. Yeah, and a lot of times, if we can say we're taking pain medication for swelling, for example. If you can decrease the swelling, that's going to be so much better for you than taking the pain medication. So be careful with your body chemistry and some of these medications. They can cause all sorts of problems.
Create more pain.
Yeah, in the long run. So homeostatic imbalances on page 489 talk about things like fibromyalgia, long-lasting, constant pain. It has to do with a lot of these pathways and receptor and perception. Phantom pain, where we're actually feeling pain in something that's no longer there. Say a limb is removed. Yeah, and you get, because you're still getting residual chemistry basically that's going on with those receptors that were there sending messages to the brain that are still being sorted out by the brain. So feeling something that's not there.
That happens in the fibromyalgia too.
Yeah, exactly. Because again,
They still receive it an hour later.
yep, they still perceive that pain because they're still getting messages from those receptors. So stimulation of visceral organs, for example, is sometimes going to cause you to perceive pain in a place that it really isn't. For example, if you're having a gallbladder attack, you might think you're having a heart attack. Heart here, gallbladder there. Yes? Not in the same place, but because of referred pain and because of the processing that's going on in your perception of that pain, you might feel it somewhere else. And that has to do with chemicals and pathways of all these different things that we're talking about. So referred pain, we feel it in different body regions. Again, heart attack, what's one of the things people talk about? When someone's having a heart attack, they feel pain where? Down their arm.
Women are different.
Yeah, women can, and again, everybody's different. We're all wired differently. So when you're having pain associated with the heart, it might not necessarily play out the way it's described in your textbook. And we definitely see that a lot more in women than we do in men. So this is sort of a map of referred pain. So when I'm having pain in these different places, I feel them other places in my body. So heart pain can be up. Sometimes people get jaw pain when they're having heart issues. Nerves. Structure and classification of a nerve. We're hitting page 490 and 491. So when we talk about a nerve, we're talking about a group of what? Neurons. So when I say nerve, I mean lots of neurons. And they're shoved into these little pipes that create nerves. So when we look at a nerve in the peripheral nervous system, it's a bundle of myelinated and unmyelinated neuron axons, peripheral axons enclosed in some sort of covering or connective tissue. When we discuss muscles, you're going to see these same exact words, but they're going to be slightly different. Instead of neurium at the end of the word, we're going to see mesium at the end of the word. But it's the same darn thing. These are coverings of the nervous system, therefore the ending is neurium. So, connective tissue coverings include the endoneurium, and what's that? Well, around the what? Because we're going to see there's coverings several different places. We're going to see coverings around the actual neurons, and then we're going to bundle them all up, and then we're going to cover them, and then we're going to cover the whole thing.
Isn't that what perineum means?
Peri means around.
So you're telling me they do the same thing?
Not quite. Kinda, but not quite. They're in different places. They're around the outside, yes. So here's what I want you to think of. And this is probably dating myself. Maybe some of you younger folks won't remember this, but when I was in kindergarten, we used to have milk and snack time. Did you have that in kindergarten? And the teacher would come out with a big brown box of straws. Yes? Do you remember this? Okay, envision the brown box of straws. All right, that would be the epineurium of my neuron. And then the teacher is going to come in and grab a bunch of my straws so she can hand them out to the classroom. That would be the perineurium, my teacher's hand covering that bunch of straws. That bunch within the big bunch, the big box of straws, would be called a fascicle, and would be covered by the perineurium. And then each of the straws has its own little covering, right? Because I don't want my teacher's fingers all over my straws. That little white paper that covers all your straws, that would be the what? Endoneurium. Okay? So we're talking about coverings around bundles. Why are these coverings so important? So this is an actual histology slide or picture of looking into a nerve, a group of neurons. So we see fascicles or groups. Can you see them? There's one here, there's one here, there's one there. So those are like little bunches of straws. Then we see the perineurium and the epineurium. Let's look at the diagram. It's a little bit easier to see. So we see the bunches, the fascicles. We see, of course, a blood supply. That's very important in all nerves because why? Yeah, we're going to get nutrients and get rid of wastes for the cells themselves. Then we have those coverings, and what's that covering around my bunch of straws called? Perineurium. The wrapper on my straw is called the endoneurium, and the box that everybody's in is in the epineurium. We good? You're going to see the same exact words for a muscle. Because a muscle, like this muscle, what's this? My biceps is actually a group of muscle cells. And they're grouped exactly the same way. So we're going to see those terms again. So you should know these terms and understand what they mean with respect to a nerve. So when we look at any given nerve, we're going to see a whole bunch of axons, right? From a whole bunch of different things, and they're all covered up and they're all separated. So most nerves are what we call mixed nerves. And what's that mean? No. No, not male and female. They can be sending messages from a whole bunch of different places. Not only sending messages, but receiving messages. So there can be sensory and motor pathways in any given nerve group. So we classify them according to the direction they transmit information. If we have a mixed nerve, we have both sensory and what? And motor. So we can actually have pathways going both ways in that same nerve group. Information coming from my hand, sensory, and motor information coming out to cause it to move. Can be in the same nerve group. We do have some that are just sensory or afferent, and they only send impulses where? To central nervous system. And then we have just motor or efferent, and they send only message from the central nervous system out. Yes? So if we have a pure sensory or afferent or motor efferent nerve, we can have different rates at which those messages are being sent. There's different types of fibers and different speeds at which message is being sent. So different fibers in mixed nerves. And what was a mixed nerve again? Sensory and motor. We can have somatic, afferent. What's that mean? Body going in. Somatic, efferent. Body going out. Visceral, afferent, visceral, efferent. Yes? Peripheral nerve classification, cranial or spinal nerves. Here's where the fun starts. You skip. Your brain is a sponge. I guess they said that. All right. So ganglia are groups of cell bodies that we're going to find on the trail of a nerve group. They're associated with afferent nerve fibers that contain cell bodies of sensory neurons. If we have a dorsal root ganglia, dorsal is what? Dorsal's back, but dorsal in respect to the nervous system and the spinal cord is going to bring sensory information, sensory somatic. That was from chapter 12. Yep. Ganglia associated with efferent fibers. Or is it going to be where? Yeah, it's called the autonomic ganglia and will be the ventral branch, exactly. Mature neurons are amniotic. Holy. I know, I should have. I probably should have.
That's what I was thinking.
Bailey's little Kahlua.
It's gotta be what it is.
Cocoa with some peppermint schnapps would be nice.
Yeah, I agree.
Amniotic, without what? Without mitosis. But if soma of damaged nerve is intact, soma means what? Body. I can repair some damage in the extensions. So that's why I was confused. They kind of skipped over that. But page 492, figure 13.5, shows us how we can actually regenerate neuron fibers as long as the cell body is intact. So sometimes, say I get a deep cut, and I used to show this movie in my lab class. It was really cool. Guy falls and cuts himself with a big piece of glass. Now, pull out the glass. I've severed a nerve. That pipeline of what? Axons. But the cell bodies of those guys are still intact. So I can, if I bring those pieces back together, I can regenerate that impulse. I can repair those damaged axons as long as the cell body is intact. Once I kill the cell body, you're SOL. But I can repair the damage there.
You have to sew the nerve back.
Yes, you have to bring the ends back together, and then you have to allow for this process to take place before the message gets sent.
Doesn't that hurt?
Yes.
I know my brother had a nerve sewn when he had a finger sewn. They messed it up a little bit.
Yeah. So axon fragments Wallerian degeneration spreads distally from the injury. It's sort of the message that the neuron sends that something's wrong and we have to start repair. Macrophages are going to play a role in cleaning up the mess. They're also going to help regenerate or reconnect that myelin sheath. Axon filaments can grow through regeneration tubes, and axon regeneration and new myelin sheath can form. So it's kind of like the steps that we talked about when we talked about forming new bone, when we repaired a fracture.
That's why blunt trauma generally doesn't heal the same as a clean cut.
There's more mess.
Like you can lose your finger and have it put back on if it's cut,
Exactly.
very clean.
And basically, what do you have to do? Bring the edges back together so that the cells can knit themselves back together and regenerate. It's the same with an axon. So the greater the distance between the severed ends, the less chance of regeneration. So we have to go to the hospital and have them do what? Bring the ends back together so this process can take place. Again, the pictures are there for you on page 492, figure 13.5, steps 1, 2, 3, and 4. Oligodendrocytes. Remember, those are the guys that made what? What are oligodendrocytes?
Myelin sheath.
That's the myelin sheath where?
Central.
In the central nervous system. They bear growth-inhibiting proteins that prevent central nervous system fibers from regeneration. We're talking peripheral nervous system, but if we do too much damage to the central nervous system, we're going to have trouble with this process as well. Astrocytes can help me with this. And some growth factors, some chemicals can help me with this. But it's not as easily done in the central nervous system as it is in the peripheral nervous system. So these are the steps. Axon becomes fragmented at the injury site. Macrophages are going to clean up the region, get rid of the dead debris, help knit together, or help to bring together that axon. And then we help to regenerate that axon as it sprouts filaments and grows through the tube and reforms those Schwann cells on the outside.
So you can technically grow nerves then.
You can grow nerve processes. See the difference?
You can't actually grow the nerve.
Once a cell is differentiated into what it's going to be for the rest of its life,
Yeah.
no. But if I could unlock the secret of cell differentiation and cause an immature cell to differentiate into a nerve cell or a neuron, then I could grow. And who are the cells that can differentiate? Yeah, and that's what stem cell research and with respect to the nervous system is all about. What is it that causes those cells to change into a neuron? So if I can have a group of those cells and cause them to differentiate into neurons, I might be able to repair some of that nerve damage. Oh, the cranial nerves. Do you have to know them? Yup. On page 493, figure 13.6, cranial nerves 1 through 12. You need to know them by name, you need to know them by number, and you have to have a general understanding of what they do for a living. This little cutesy saying is what we call a what? A mnemonic. Okay? That's one. Make your own. You'll remember it better. So there's 12. I'll give you the first two right off the bat. How many noses do you have? 3. 1. One nose. Cranial nerve 1 is your what? Olfactory, sense of smell. How many eyeballs do you have? 2. Cranial nerve 2. Optic, your sense of sight. There you go. So if you look on page 493, and then it's going to skip to page 500. They are listed for you, all 12 of them. Optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, cranial nerve, 8, glossopharyngeal, vagus, accessory, and hypoglossal. Those definitions, know them. When we dissect the sheep brain in lab, pay attention to try and find as many as you can. It's going to be very difficult because you have to take off your dura mater. And unfortunately, when you take off your dura mater, you might rip off a lot of these very fine cranial nerves. But we call them cranial nerves because they're in the, there you go, they're in the cranium. And they're kind of lined up in order. This is going to give you some basic information, the bottom of that diagram I mentioned before, on their function, sensory and motor. And what's PS fiber? By the way, parasympathetic, which is what? Slow things down. So, rather than bore you the living daylights out of you by going through exactly what you have written, you need to know them all. Optic, olfactory, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus. Vegas is kind of a place we go away, right? Vegas, we go away to Vegas, right? Vagus, cranial nerve 10, is pretty much the only one that's going to go away with respect to impulse from this, the cranium, yes? So it's going to control things other places like the thoracic region, okay? So think of Vegas, we go away from the head to have fun. So when we look at the information that's being sent via the vagus nerve, we're not looking at just things in the cranium, we're looking at lungs and heart and digestive organs. We're getting messages from there as well and sending message. Away from the head. Accessory, hypoglossal, and cranial nerves. Composition.
So obviously the vagus nerve is going to be large.
It's fairly good sized, yeah.
Oh, it's going to go to all that type of stuff.
Yeah. If you look at that diagram, I'm not going to go back to it, but look on page. If you look on page 493 and you find vagus, it's quite thick. Spinal nerves. You have 31 pairs of spinal nerves. And they're pretty much going to follow the same numbers that you already know with respect to the bones that they are associated with, except for cervical. How many cervical vertebrae do you have? 7. How many cervical spinal nerves do you have? 8. Why? Top, bottom. Yes? So since cervical happens to be at the top of the stack, those guys are going to have 1 extra. Everybody else follows the bone rules. 12 thoracic thoracic spinal nerves. 5 lumbar. 5 sacral. Remember we talked about the bone itself? It was the fusion of 5 separate bones that form the sacrum. Remember that number when you think of how many sacral spinal nerves you have. You have 1 little coccyx, that word. There's no way I'm going to be able to say that word today. All right? So, 31 pairs, 7 cervical vertebrae, yet 8 pairs of cervical spinal nerves. This diagram on page 501 shows us how they are numbered. Spinal roots. Each spinal nerve connects to a spinal cord via the spinal root. So think of the wires in your house running from that switch when I flip it on to the source and where processing is taking place. So there's a ventral root that's going to send messages where? Motor. And a dorsal root that's going to bring messages where? Yeah, sending up or ascending. And I hate to end here, but I'm going to end here. There's not much left. You might.
Why the cost of zero is considered C0 instead of C1?
I don't know. I don't have the answer to that
Why
question.
cervical? Probably cervical.
Oh, probably. You don't want to get them messed up. All right, so we will pick up on the dorsal root of our spinal nerves on Tuesday. I just want to tell you one thing before I stop my recording, so everyone hears it, including the people that didn't come to class today. Very soon in your future is your next exam. I highly suggest to you that you spend quite a bit of time this week on studying 11 and 12 and 13, okay? Because before you know it, and it's going to be very quickly, that test will be upon you. Use your interactive physiology, use your mastering of A&P. You will be crazy if you don't.